Hello, good morning, and welcome to today's webinar from Takara Quantec on our next generation tools to clone, express, and purify proteins to really speed up your protein expression and purification workflow. My name is Malti Rahman, and I work as a product manager at Takara Bio Europe, and I'll be presenting today's webinar. There will be time for questions at the end, but if you have questions during the webinar, please just type them into the chat or question box. This webinar is also being recorded, and we will send you a link to the recorded version as well as sending you a copy of today's presentation in a few days. So let's begin the webinar. So before I go into the different technologies that we offer for protein expression and purification, I just wanted to go through a typical protein expression and purification workflow that most researchers follow, and where we offer tools to ease and speed up this workflow. The first step within protein expression and purification is to clone your gene of interest that encodes your target protein that you want to express into your specific protein expression vector. Normally most researchers do this via traditional ligation-based cloning, but here I want to introduce you to our next generation infusion HD cloning technology, which is innovative and can really help you to speed up this step. After generation of your protein expression construct, the second step is then to express your specific target protein. And normally people express proteins as a recombinant protein, usually with a tag such as a his tag. And we offer a wide choice of different systems for this step, and I'll talk about each of these briefly later in my talk. So after expression of your his tag protein, the next step is to purify this his tag protein. And in fact, we have a great new technology known as our next gen Capture-M technology for easy purification of his tagged proteins. However, if you still prefer to use more traditional resin-based methods, then we also still offer our Talon and his 16 nickel resins for his tag protein purification, and I will talk about these as I go through my talk. Okay, so as I just said, the first step of any protein expression and purification workflow is to clone the target protein into a specific protein expression vector. So as you know, cloning usually represents the first step of most molecular biology experiments carried out in labs today, but it's not usually the main focus of the experiment, but rather a first essential step on the way to running your actual experiment and achieving your research goals, i.e. publishing your paper. Now, most people still use traditional ligation-based methods for cloning, which can be very time-consuming when things don't work well. And this has happened to me many times where I've spent many months on one construct, and this can really hold you up with the rest of your experiments. But with infusion cloning, we believe that we found a faster, more highly efficient, and just a better way to clone. So imagine a cloning system that will allow you to clone both single and multiple inserts in a single reaction, that will allow you to do site-directed mutagenesis, as well as the cloning of C and N terminal tags. Really a cloning system with no limitations. Well, we believe that we have such a system. This is our next generation Infusion HD Cloning Plus system. And this is what I'm going to talk about in my next few slides. So what's so great about next generation Infusion HD cloning technology? We really feel that this is a really better way to clone compared to traditional ligation-based methods. And the beauty of infusion lies in the fact that it can be used to clone any insert into any vector at any locus. And this cloning is also seamless, directional, and high throughput ready. So the most important feature of infusion is that it's sequence independent, so it can be used to clone any insert into any vector at any locus, and you're really not limited by available restriction sites, which is a major limitation of traditional ligation-based methods. Infusion also allows you to directly clone into your destination vector, which can be any vector, so you don't need to do numerous sub-cloning steps, unlike traditional ligation-based methods. Infusion is also seamless and directional. So this means that no extra bases are added to your insert sequence unless you want to add them. So this can often be an issue with other cloning methods such as traditional ligation-based methods and also gateway recombination cloning, because often these add sequence scars, which in the context of protein expression and purification can add additional amino acids to your target protein, which can sometimes alter the function of the protein. But with infusion you can avoid this problem. It's also possible to use infusion for several nice applications such as multiple fragment cloning, which is possible in a single reaction, as well as for carrying out site-directed mutagenesis, including generation of substitutions, deletions or insertions, again in a single step. And really, it can be used for all your cloning experiments, and all the cloning experiments can be done in a single step. So you might be asking yourself, how does this infusion enzyme actually work? Well, the first step involves generating your linearized vector at the desired locus that you want to clone your gene of interest into. So this can be achieved either via restriction digestion if you have suitable and available restriction sites, or by inverse PCR if you want to be completely restriction site independent. You just need to know the 15 bases found at either end of your linearized vector, shown in red and yellow here, because the same 15 bases need to be added to either end of your insert that you want to clone. And this can be any insert. You're not limited by the sequence of the insert. So you generate this insert via PCR using gene-specific primers that also include the 15 base pair overlaps, which are homologous to the ends of your linearized vector. So once you've generated this, you then simply incubate the insert and vector with the infusion HD enzyme for 15 minutes at 50 degrees C. And what the infusion HD enzyme does is it fuses the insert to the vector at these 15 base pair overlapping regions. This infusion reaction is then transformed into E. coli, where any gaps get repaired and you get positive clones containing your recombinant vector. So really, because you don't treat your PCR insert in any way, you're just not limited by the sequence of your insert and your vector. And it's really a very fast and accurate method. So now I just want to talk a bit more about how the actual infusion HD enzyme works. So as I mentioned before, the first step is to linearize your vector. So if you imagine that this is a linear vector, so this black region is missing here, and you have the 15 bases found at either end, shown in red and blue here. Now the same 15 bases have been added to either end of the insert via PCR. So when you actually incubate the insert and vector with infusion HD enzymes, what the infusion HD enzyme does is it creates single-stranded regions at the ends of the insert and at the ends of the vector, which then allows the insert to fuse to the vector at the revealed 15 base pair homologous regions. And this is spontaneous annealing. And it's only upon transformation into E. coli that any gaps get repaired and you get generation of positive colonies containing your recombinant vector. So let's watch this in real time. So when the insert and vector are incubated with the infusion enzyme, it creates single-stranded regions at the end of the insert and vector, which reveals these spontaneous overlapping regions, allowing the insert and vector to fuse together at these 15 base pair overlapping regions. So after this step, this is then transformed into E. coli where any gaps get repaired and you get positive clones containing your recombinant vector. So it's really as simple as that. So remember, this is ligation independent and it's also not exactly recombination. So what are the primary advantages of using Infusion. Firstly, it's a really great cloning system which allows researchers to be very successful in all their cloning experiments. This is because we've invested over 10 years of progressive research into perfecting our product. It allows you to clone any insert into any vector at any locus. And this cloning is seamless, so no extra bases are added unless you want to add them. Infusion also has an excellent performance. We obtain over 95% positive recombinant clones or cloning efficiency across a wide range of insert sizes ranging from 20 base pair oligos to 15 kb inserts. So really each time you just need to screen 2 colonies and both should be positive. Infusion is also fast and accurate. It's also simple and powerful, so you're not limited by the size or number of inserts. And it's also one system that can be used for multiple applications, as I'll discuss later. So this slide here illustrates the efficiency and accuracy of Infusion. So this graph here has plotted the total number of colonies against the insert size. So as you can see, as you clone larger fragments, i.e. going from an 8 kb to a 12 and 15 kb insert, the total number of colonies does drop as would be expected. But however, the percentage of positive clones as assessed by colony PCR and sequencing remains high. So we obtain 90 to 100% correct clones for all these 3 different fragments. And you can achieve the same efficiency and accuracy using either our Infusion HD liquid systems, which allow customizable reaction volumes and are scalable for high throughput applications, or you can use our Infusion HD EcoDry system, which is a lyophilized version of our Infusion enzyme. And this is great because it can be stored at room temperature. So if you're short on freezer space or if you want to be more environmentally friendly, this could be a great option for you. Now, we know that sequencing is the gold standard for measuring the accuracy of cloning. So here in this slide we've shown some sequence data of positive clones obtained with Infusion. As you can see, the cloning is always directional and it's accurate. There are no errors in the insert sequences due to the use of our high fidelity cloner and Purify polymerase that I'll talk about later. But also you can see that the cloning is seamless, so there are no extra bases added between vector and insert. And as I said before, this is particularly important when generating protein expression constructs because you don't want to add any extra bases or additional amino acids to your target protein because this could potentially change the function of your protein. So with Infusion you can avoid this problem. Another great thing about Infusion is its versatility and the fact that it can be used for several different applications, including single fragment cloning, cloning into large vectors such as cosmid vectors, multiple fragment cloning, and deletion insertion mutagenesis, substitution mutagenesis, as well as for the addition of small N or C terminal tags. I won't have time to talk about all these applications, but I'm going to focus just on multiple fragment cloning and site-directed mutagenesis. And really, Infusion is such a powerful cloning system that you can design any construct and Infusion can deliver this construct to you. Now, multiple fragment cloning is a great application of Infusion because it can be achieved in a single step and you're not limited by the sequences of your insert or vector. And remember, this is really not possible with traditional ligation-based cloning because you're often restricted by the available restriction sites, and also often you need to do several rounds of subcloning before obtaining your final construct. But with Infusion, multiple fragment cloning is very easy. Okay, so how does it work? So as always, the first step involves generating your linearized vector either via restriction digestion or via inverse PCR. The next step involves generating your various different inserts. So here we've just shown 3 inserts as an example. Insert 1 is shown in red, insert 2 in blue, and insert 3 in green. So here primer design is crucial because you need to make sure that each of your inserts either carries a 15 base pair overlap to one end of the linearized vector or to the adjacent fragment to which it will be joined. And we have an online primer design tool to help with this. And in fact, we found that 20 base pair overlaps works better for multiple fragment cloning compared to 15 base pair overlaps, which is why we've developed a specific multiple fragment cloning protocol which recommends 20 base pair overlaps, and this will be coming out soon. So you just need to remember in the primer design that either the primer has to carry a 20 base pair overlap to one end of the vector or to the adjacent fragment. So let's go through this example. So here, fragment 1 shown in red here, the forward primer has a 20 base pair overlap to one end of the linear vector, and the reverse primer has a 20 base pair overlap to the second fragment shown in blue here. And similarly, the reverse primer of the second fragment has a 20 base pair overlap to the third fragment shown in green here. And finally, the reverse primer of the third fragment has an overlap with the other end of the linear vector. So what this means is when these inserts and this linearized vector is incubated with the infusion enzyme, the infusion enzyme, as before, creates single-stranded regions at the ends of each insert and vector, and it allows them to assemble at the homologous overlapping regions. So all the fragments spontaneously anneal at the 20 base pair overlapping regions. So effectively the 3 fragments are cloned as a single fragment via infusion. So this is amazing because this allows multiple fragment cloning in a single reaction. And don't just take our word for it, we have a nice paper that illustrates this application of infusion. So here I just wanted to show you some in-house data for a 3 fragment cloning that we did in-house. So here 3 PCR inserts of 1.1 or 0.7 kb, i.e. MBP, Prof12 and ACGFP1 were cloned into linearized PUC19 using infusion to assemble a final construct of 5.2 kb in size. Now, as you can see, after the infusion reaction we obtained 89 colonies in total, only 1 on the negative control, and when 26 out of the 89 was further screened by colony PCR and sequencing, we obtained 100% cloning efficiency, i.e. 26 out of 26 were correct. So this is a great result. And I should mention here that with multiple fragment cloning, as you clone more and more fragments, the number of colonies you obtain drops, but the percentage of positive clones remains high, and you just need 1 clone in the end. Now another way that you can make multiple fragment cloning via infusion even easier is to generate your various inserts as synthesized DNA fragments, as this customer from MIT did. So first he tried to synthesize a 3 kb gene, but he wasn't able to do this due to strong secondary structure within the gene, and he didn't want to introduce restriction sites in the middle of the gene. So instead what he did was he split the gene into 2 and generated 2 fragments by DNA synthesis, but including the 20 base pair overlaps required for infusion cloning, and then just used infusion as normal to seamlessly join the 2 fragments. This is a really great way of speeding up your multiple fragment cloning workflow. Another great application of infusion is site-directed mutagenesis. So you don't need a separate kit for mutagenesis if you have infusion in your lab. So how does this work? This works by combining the power of inverse PCR with infusion. So the first step involves choosing the site to be mutated. So if you have a vector that already contains your gene of interest and you just want to specifically mutated a few bases, you just pick this region. So this is shown in yellow here, And then you simply generate inverse PCR primers, where the 5' end of each primer contains the bases to be mutated, shown as 3 X's here. But they also contain the 15 base pair overlap. So in this case, the base pair overlap is with each other. So then you generate your inverse PCR product using this primer. So you end up with a linearized vector which contains the mutated bases and the overlapping regions. And then you simply add the infusion enzyme which recircularizes the vector at these overlapping regions. So you obtain your new recombinant vector containing your mutated bases. So this is a really nice application of infusion, and you can read more about this in our Nature Methods app note. So now I want to talk to you about our latest formulation of infusion, known as Infusion HD Cloning Plus, which is a complete cloning solution, and we believe it's the cloning solution for all the cloning experiments in your lab because it allows fast, efficient, trouble-free cloning. So what comes inside the box? The Infusion HD cloning enzyme. We also provide a high-fidelity CloneAmp Hi-Fi PCR premix to generate error-free PCR inserts, as well as an insert purification kit or cloning enhancer for purification of your inserts. And finally, we also provide a highly competent stellar cells for high out-of-the-box success rate. But equally, because we believe in providing a whole product solution, even outside of the box, we provide expert technical support to help customers achieve successful infusion cloning. We're also dedicated to our customers' success, and we're continually trying to improve and innovate our products. And in fact, our Infusion HD Cloning Plus is a value packaging, because if you were to purchase these components separately, it would be much more expensive. So let me just talk about two of the optimized subcomponents that we provide in Infusion HD Cloning Plus. I first want to talk about our CloneAmp Hi-Fi polymerase. Why do we provide this in Infusion Plus kits? This is because it's a very high fidelity enzyme that has a very low error rate, just 12 errors per 542 kb, and in fact only one error per 45.2 kb on a challenging GC-rich template. And as you can see, this error rate is much lower than competitor polymerases on the market. And this is ideal for cloning because you need error-free inserts. And moreover, CloneAmp Hi-Fi is very fast. It has an extension speed of 5 seconds per kb, allowing fast cloning. We also provide our stellar competence cells in our Infusion Plus systems because they're optimized and they've been optimized for infusion cloning, and they're ultra high efficiency. So as you can see, when we compare different competence cells, we obtain the most number of colonies with stellar cells using infusion cloning. So although you can use other cells such as HB101, JM109 or DH5 alpha cells, and the number of colonies or the cloning efficiency isn't as high. And moreover, certain strains don't work as well at all, such as top 10 and DH10 beta cells. So we don't recommend using these cells with infusion. But equally, these are high quality cells that can be used for routine cloning experiments. And similarly, even CloneAmp Hi-Fi can be used for routine PCR experiments, so not just infusion cloning. Now, I just wanted to finish with an example of how we're dedicated to our customers' success. We will soon be launching an Infusion mobile app, which has a colony counter and in-gel DNA concentration modules. So here, this app will allow you to take a picture of your plate or your gel with your smartphone, and then the app will count the number of colonies on your plate, or it will be able to measure the concentration of DNA in your band. So it's a really nice app which should be coming out before the end of the year. And don't just take our word for it. We have many papers that have successfully used infusion. In fact, over 200 publications now. And if you want to find out more information, you can visit our cloning resource portal, which is a valuable source of information for infusion cloning and our other cloning products. Or you can email our tech support address too. So I've talked in detail about the first step, which is to clone the protein expression construct. The second step is to then express your target protein. So for this, we provide a wide choice of expression systems, including bacterial, insect, mammalian, and human cell-free systems. And as you can see, as you go from bacterial to human, the number of post-translational modifications increases. So how do you know which expression system to choose? Well, this really depends upon your application and your needs. So this is summarized in this table below. And although I won't talk about all these systems in detail today because there isn't time, I will just touch briefly on our different bacterial systems. So, as I said, the first choice system that we recommend for people who are new to protein expression is our PET Express and Purify E. coli system, which is simple and fairly high yield, so you get up to 1 gram per liter. But if you have proteins which are a little bit more tricky or temperature sensitive, then you could consider using our cold shock expression vectors, which allow expression of the protein at 15 degrees C. But however, if you have a more complex protein, which is toxic, maybe our single protein production system would be ideal for you. Although this is a little bit lower yield, it's good for tricky proteins. Alternatively, if you want a secretory protein expression system that gives you high yield, then you could consider using our Brevibacillus or B. satilis systems for this purpose. B. satilis gives you the highest yield at 25 grams per liter, but Brevibacillus is also good with a yield of 4 grams per liter, and both allow certain post-translation modifications such as the formation of disulfide bonds. And with Brevibacillus, secretion is optional, whereas with B. satilis, it's optimized. And again, Brevibacillus is also good for temperature-sensitive proteins. However, if you're looking for a system that allows many post-translation modifications, for example, if you have a complex eukaryotic proteins, then maybe our baculovirus backpack system would be the best option for you because it allows many post-translation modifications. And again, the protein is secreted into the media and allows temperature-sensitive protein expression. And finally, if you have a very toxic protein, but yield isn't so essential, then you could use our in vitro transcription and translation system, our human cell-free system, which again allows many post-translation modifications and temperature-sensitive protein expression, but the yield isn't so high. So this really depends on your needs. So I just want to touch now on all the different E. coli and bacillus systems that we offer. So our first choice system that we always recommend to researchers is our PET Express and Purify system, which is based on the PET vector system and allows IPTG-inducible expression of your target protein. And you get strong expression from the T7 LAC promoter. And actually we provide both N or C-terminal His tag vectors. So once you express your protein, you can then easily purify it using the His tag. So let me just talk about how this system actually, or kit, works. As I said before, the first step involves generating your protein expression construct. So you need to clone the gene of interest into the PET vector. And here you can again either use infusion if you use our infusion ready systems, or you can use traditional ligation-based cloning methods. It's your choice. But after this step, you then add IPTG, transform it into E. coli and add IPTG for induction of your protein. Now, how does this IPTG induction work? So normally the strain that you use is your BL21DE3 based E. coli strain. So here, in this strain, the T7 polymerase expression is inhibited by the LAC repressor. So by IPTG induction, the lac repressor dissociates, allows T7 RNA polymerase expression, which then also binds to the T7 promoter within this vector and leads to high level expression of your target gene from the T7 lac promoter. Because again, IPTG induction removes the lac repressor. So once you've expressed your his-tagged protein, with our PET Express and Purify systems, we also provide a choice of our his-talon or his-60 nickel resin, so you can then purify the his-tagged protein. So really this is a complete system. Or, as I'll mention later, you can combine it with our Capture-M mini prep kits for easy purification of his-tagged protein. Now, if you have a slightly trickier protein, which is not so soluble, then you could consider using our P-cold expression vectors instead as an alternative to the PET Express and Purify kit, because it allows protein expression at low temperatures, specifically 15 degrees C, making use of our cold shock promoter. And what this means is this suppresses the synthesis of most other proteins in the host cell due to the low temperature induction, and ensures that the target protein represents up to 60% of all expressed cellular proteins. And in fact, due to the low temperature, this also often increases the solubility of the target protein. And again, two of the vectors that we provide allow his-tagged protein expression, which can then be used with our downstream Capture-M or his-60 nickel talon resin based purification systems. Now, for very tricky proteins, you could consider using our elegant single protein production systems, which just allows you to express simply the target gene. So with this system, 90% of your total cellular protein will be your target gene. So how does this work exactly? This is a two-vector system. So firstly, a modified P-cold vector is used to express the target gene. In fact, this P-cold vector has had all the ACA sequences removed, and even your target gene needs all the ACA sequences to be mutated. And then you also co-transform the PMAS-F vector, which encodes the MAS-F endoribonuclease. So this nuclease specifically cleaves single-stranded RNA, i.e. single-stranded host mRNA molecules at ACA sequences. So what happens when you co-transform both of these vectors is that all the host mRNAs containing ACA sequences will be cleaved by MAS-F, whereas your target mRNA will not be cleaved because you've already mutated all the ACA sequences. So this results in high-level expression of just your target protein. This is a really nice, elegant system, but it requires a lot of work to mutate the ACA sequences in your target gene. But if you have a very difficult protein, you could consider using this system. Now I want to talk about our Brevibacillus system, which allows secreted protein expression, and often this gives better results than E. coli. This is because you can get some post-translation modifications such as disulfide bond formation, and also it's good for toxic proteins because it doesn't stay in the cell too long, it's secreted quickly, and often you get more native active proteins compared to E. coli. And in fact, with our new system we have two vectors, so either you can choose a secreted vector or the intracellular vector, it's your choice. And again, you can either use classical cloning or our new BIC cloning system to generate your expression construct. So how does a BIC cloning system work? It's similar in a little in concept to infusion, but it's not exactly the same. So here, instead of using a shuttle vector, instead of first cloning in E. coli and then transforming into Bacillus, you can directly clone and transform in Bacillus. So here, again, you generate your target gene with 15 base pair overlaps to either end of the linearized PBIC vector. So the insert and vector is then transformed into brevibacillus cells, and here the insert's cloned into the vector via homologous recombination. So it's a little bit different to infusion. But this is nice because this way you don't have to do a two-step cloning procedure. You can just directly clone into brevibacillus. However, the best system for secreted protein expression, especially if you want a very high yield, is our B-Satillus system. And this is because this is a safe organism which allows you to push secretion to a maximum. And in fact, what our vector system allows you to do is first clone your gene of interest or protein of interest into the PBES vector, into the multiple cloning site. So this can be achieved via infusion or ligation-based cloning. This is your choice. After this step, you then cleave your PBS target gene vector with MLU1 and ECHO521, which removes the signal peptide sequence. And then we provide a library or 173 different types of signal peptide DNA. So once you've removed this and you have your linearized vector with your target gene, you then use infusion to generate a library of different PBS vectors containing your target gene but different signal peptides, i.e. 173 different signal peptides, to allow you to screen which signal peptide gives you optimal secretion. So this is a really nice step which combines our vector system with infusion cloning. So in summary, E. coli is the first choice that we recommend for a high yield of target protein because it's simple, it's a complete system, and it allows fairly high yield expression. But if E. coli fails to produce active protein, then you could consider using our P-cold vectors for low temperature expression or our SPP system for single protein production. Or alternative brevibacillus and B-Satillus for secreted protein expression if you need high yield protein or active protein or protein which is normally toxic to cells, but in this case is secreted quickly into the medium, so it can be easily expressed using these bacillus systems. However, if you need more post-translational modifications, then our baculoviral backpack system, though it's a bit more complicated to set up, it would be ideal for this application because it would allow you to produce high amounts of active protein containing the various post-translation modifications that you require. And finally, you can also consider using our human cell-free expression system, which is an in vitro transcription and translation system if yield isn't so critical, but yeah, you just want to easily express a protein which is normally toxic. This could be an option, but again, costs will be higher. So now let's move to the third step of the protein expression and purification workflow. So far I've talked about cloning and expressing the protein. Now let's talk about purifying target proteins. So here I'll specifically talk about our protein purification products for the purification of His-tagged proteins, because most researchers use His tags because it's a very short tag which doesn't normally affect the function of the protein, and it's usually well tolerated. So we provide both resins for classical protein purification. So in this case we provide our His-60 nickel and Talon cobalt based resins. So His-60 nickel is ideal for high yield, but although it's reasonably pure compared to competitors such as nickel NTA from Qiagen, the purity isn't as high as with our Talon cobalt resin, which is unrivaled for purity, but the yield is lower than the His-60 nickel. But in both cases you get low metal ion leakage, and we provide a choice of formats for different purification scales, i.e. bulk resins, magnetic beads, FPLC cartridges, which can be used in actor systems, and gravity and spin columns. So let me just briefly talk about the differences between Talon and his 60 nickel. So applications that require the highest purity, we would recommend using Talon. For example, if you're doing crystallography or structural investigations or functional assays, then Talon would be the best option because it gives you the highest purity and has a binding capacity of 20 mg per mil, so slightly lower yield than his 60 nickel. But it allows purification under native conditions, such as under beta-mercaptoethanol, and you can also do a pH elution. But normally we recommend elution using 150 millimolar of imidazole. Whereas with our His 60 nickel superflow resin, we recommend this for applications that require large quantities of protein, such as protein labeling or immunization studies or animal studies. So although His 60 nickel has a good purity, it's not as high as Talon, but the binding capacity is higher, so you get 60 mg per mil. So yield is higher with His 60 nickel, but purity is better with Talon. Again, it also allows purification under native conditions, although it's not as good as Talon in terms of performance under beta-mercaptoethanol, but this is not so surprising as it's a nickel-based resin. And again, purification is usually carried out using imidazole, so this time we recommend using 300 millimolar imidazole. And as I said before, we provide various different formats, including bulk resin. So this is usually the most popular format. So here you just buy the resin from us and you can pack it into your own columns. Or we provide cartridges which are pre-packed. So these are ideal for use in FPLC, using machines such as ACTA. Or we provide our gravity pre-packed columns. So here protein is just purified via gravity flow. So it's up to you. But we provide all the different options. Now I want to end the final part of my seminar talking about our brand new CaptureM next-gen protein purification kits, where simplicity meets high performance. So what's so great about our CaptureM technology, and why do we believe that it's a next generation His-Tag protein purification technology? This is because unlike resin-based methods, CaptureM is very fast and it's very easy to use. So you don't need to spend hours purifying proteins in the cold room. Why is this? What makes it so unique? The unique feature of our CaptureM kits is that we provide either spin columns or plates. So we have Mini Prep, Maxi Prep and 9612 versions, which all contain high capacity nickel-based nylon membranes instead of resin. So almost all other commercial providers provide resin-based purification systems, whereas we provide a membrane-based purification system which is more robust and it's more rapid and allows purification at room temperature. In fact, you can purify His-Tag proteins within 5 minutes using our Mini Prep kit or 15 minutes using our Maxi Prep kit. So there's no other provider on the market which allows such fast purification, and moreover allows the purification of highly concentrated proteins, and also is compatible with many different lysis buffers, and in fact can be used for both native and denaturing protein purification, and tolerates common additives such as EDTA, beta-mercaptoethanol. So with traditional resin-based methods, often customers may need to do a buffer exchange before the actual purification step, but because our CaptureM technology can tolerate common additives, this is not necessary, saving you a lot of time. So it's really as easy as this picture shows here. You equilibrate your column, you bind your clarified lysate, you wash, you elute, and you're done. This is within 5 minutes for Mini Prep kits and 15 minutes for Maxi Prep kits. Okay, so what's so different about the resins? What's so different about our CaptureM membrane-based technology compared to traditional resin-based technology. So, for example, most resin-based technologies are high capacity, so they have a large internal surface area and large bed volume. So this is great because it allows you to obtain a high yield of protein, but because of the large bed volume, you get a slow diffusion of macromolecules through the pores, so it's very slow, resulting in long separation times. And often impurities are also trapped within the high bed volume. Now, also, traditional membrane pores or traditional membrane-based technologies make use of low-capacity membranes, which have a small internal surface area, which allows rapid flow but also means a low yield. So this is also not great. Whereas with our Capture technology, we have high-capacity membrane pores. So the high capacity shown, this is the edge here, allows a large internal surface area and therefore a high yield, but still rapid flow due to the small bed volume. So this is what makes our technology unique, because you get fairly high yield pure protein and you have a fast purification method.